Today I'll be talking on the disposal of contaminated culture plates and maintenance of the stock culture. So the main purpose of this disposal of contaminated culture is to lay down a procedure for proper disposal of contaminated materials to ensure all media containing microbial growth are disposed of properly. So it is likely that the inoculated culture media will contain high number of microorganisms which constitute a potential hazard. They must therefore be disposed of safely by approved methods. So infected specimens and inoculated culture media should be handled by a trained personnel only. Any culture plates used must be safely disinfected or sterilized. It is suggested that the decontamination should be carried out by autoclaving at 121 degrees centigrade for 60 minutes. So after autoclaving, media may be disposed or maybe disposed of through normal waste disposal or incineration procedure. But however, media containing selenite should be disposed of as a special waste. So in that case, if any doubt as to how to handle or dispose of any specific medium, consult the local waste disposal authority. And also, before disposing of the petri plate or the dishes in the trash, the bacteria should be destroyed. So to dispose the used culture plates, the following steps should be taken. You have to take all the disposable material into an autoclave plastic bag, then autoclave the media plates at 121 degrees centigrade for 30 minutes at 15 lbs pressure, then drain the content of the plate into the sink under the running tap water, then soak the empty petri plate for 1 hour in 2.0% saffron solution, then wash thoroughly with 0.1% tipol solution. Or um, or if you have limited time to dispose the plate, you can always pour a small amount of household bleach over the colonies while holding the dish over the sink and dispose it when you get time. So this is the procedure to dispose of the contaminated plate. Next, we will move on to the maintenance of the stock culture. So proper maintenance of quality control strain is essential for ensuring the acceptable performance, as effective maintenance of stock culture is essential for quality control. um, for the quality control method validation and research purposes. And also most strain produced by microbiologists can also, like, it can be used for up to months after reconstitution when correctly maintained. But um, but repeated subculturing even like may eventually lead to contamination, loss of viability, and genotypic or phenotypic changes. Phenotypic changes, and also the freeze drying and cryogenic storage are preferred but may not be practical for the smaller laboratories. And the cryo protectant bits allowed routine labs to maintain a stock culture collection simply at a low cost. So um, talking on the storage of the reconstituted microorganism, most quality control microorganism can be maintained on non-selective agar plates or slant for up to 4 weeks at room temperature or in the refrigerator. And also the fastidious or microorganism which have shorter survival periods than aerobic bacteria, these strains need to be subcultured every few days. For example, like streptococcus pneumonia and Neisseria gonorrhea need to be subcultured every third days. So this is the storage of the reconstituted microorganism. Moving on to the typical stock culture collections. This typical stock culture collection may contain isolate that falls into one or more of the following categories, like reference strain for quality control for culture media and method, isolate used in the preparation of inoculated samples and specimens for quality control and training purposes, then reference strain for the development and validation of new methods, pathogen and spoilage microorganism related during routine testing or in the investigation of contamination problems, culture used in microbiological assay, and isolate required for the research purposes. So these are the typical stock culture collection, which may contain isolates. So this is all about the disposal of contaminated culture plates and the maintenance of the stock culture.